Aloha and welcome everyone. It's 12 o'clock and we're going to go ahead and get started. Thanks so much for joining early and on time. We're really excited. We have 3 great talks lined up today for the Science by the Sea seminar. Hopefully you can see the flyer that we have up that shows our speakers for today and their talk titles. A reminder that we are recording the seminars and posting them to the Hawaiʻi Sea Grant YouTube channel. There are a number of folks who aren't able to join us in real time who are hoping to watch the recording in future weeks. I'll start by introducing myself. Aloha, I'm Maya Walton. I'm the Assistant Director for Research and Fellowships here at the University of Hawaiʻi Sea Grant College Program. And we're so excited to welcome you to the Science by the Sea seminar. Like I was saying earlier, we have 3 great talks from 3 really impressive graduate student researchers who are supported by Sea Grant and by PI-CASC, the Pacific Islands Climate Adaptation Science Center. So in addition to pursuing graduate degrees, conducting this research, the graduate fellows that you're going to see present today are also receiving training from Sea Grant in science communication. And we ask them to do 50 hours of community outreach each year that they're supported. So we're going to hear from 3 speakers today. And then first, we're going to hear from Sea Grant graduate fellow Brytne Okuhata. Her talk is titled "A Wasteful Situation: Predicting Wastewater Transport to the Coast." Our second talk is from NOAA Fisheries Sea Grant fellow Devynn Wulstein, and her talk is titled "Taking a Leaf from the Plant Playbook." And then our third talk is from Sea Grant graduate fellow McKenzie. Her talk is titled "Hunting Coastal Springs: The Search for Coastal Contaminant Sources." Our format today is in between each talk, we'll take 1 or 2 questions from the audience. Please feel free to enter your questions into the chat as we go along. We hope you guys have lots of questions for our speakers. And I think that's it as far as logistics. Whenever you're ready, Brytne, the floor is yours.
Okay, and everybody can see my screen.
Yes, we can see your slide. It looks great.
Okay. Hi, everybody. My name is Brytne Okuhata. I'm a grad student in the Department of Earth Sciences here at UH Manoa. Today, I'm going to be talking about a project that I've been working on for a few years. We're essentially looking at the groundwater in West Hawaiʻi, specifically Kailua Kona area, and how wastewater can be transported from our development areas into the coastline. And then I also want to acknowledge a ton of different people that are involved in this project, which are listed down here. These are all of my collaborators, advisors, co-authors, so this wouldn't be possible without them. So our study area is the west side of the Big Island, specifically the Keauhou Aquifer, which is highlighted in green here. This area is of interest because it's being developed pretty heavily nowadays. And it's also a very culturally significant area along the coastline. And so stakeholder and landowner needs directed us towards this specific area of the island. So if we zoom in on this area, I'm showing you the locations of pumping wells right now. So the orange squares represent the pumping well locations that were drilled relatively earlier on because they're near the coastline, they're shallower, easier to drill. But as they started to drill further upland, we got into this red marker area. And they noticed that the aquifer up here was very productive, which is why you have this like line of wells that are drilled. And these wells were only drilled in the early 1990s. So they're pretty new wells, they're young wells. And interestingly, when you look at the water levels in all of these wells, you see that along the coastline, we have pretty shallow water depths. They're typically under 2 meters above mean sea level elevation. So you see all of the blues and the green colors. But then when you get into those high level wells that they drilled in the 1990s, you jump up to about 30 or over 100 meters above mean sea level. And so there's this pretty distinct divide between these shallow wells and these very high level wells. And so we call this the high level, the high-low divide. And the wells that are towards the right are coming from the high level aquifer, which is believed to be a very productive aquifer because of how high the water level is. There's a lot of water that could potentially be pumped from this area. And about 25% of the potable water that supplies the Kona area is pumped from these high level aquifers. And so I think a few years ago, a couple of these wells broke down at the same time. And so they kind of went into panic mode because they had to make sure you had to conserve a lot of water because they weren't able to pump as much with those wells down. So we know how important this aquifer is to them. And as I mentioned, this area is also very culturally significant. So this yellow area that I've highlighted along the coastline, those are approximate areas of where there might be an ancholine pond as determined by NOAA and TNC. And so the ancholine ponds, for those of us who don't know, are these ponds of water that are very close to the coastline, but not like directly attached to the ocean. And it's a mixture of both saltwater and freshwater that's combined in these ponds. And so it creates a very unique ecosystem and environment for a lot of species that are important to us. And one of those is like the opaiula, which a lot of us are looking into right now. And so we want to make sure that we are developing and pumping sustainably so as to not impact these ponds along the coastline. And furthermore, because this area is very developed, we have a lot of cesspools. So what I'm showing in the gray dots are all of the cesspools that are located in Keauhou. There's basically one cesspool per home, sometimes more if you have like a big building or office space. And it's only going to increase with more development, but now we're not allowed to put cesspools. We have to think of different alternatives, such as like septic tanks or ATUs. But people are very concerned that all of the wastewater that goes into these cesspools leach into the aquifer system, and then they eventually flow down to the coastline where we have all of these ancholine ponds that are very culturally and economically important. And so we want to be able to investigate like how future development scenarios, how future climate change scenarios might impact this coastline and how our pumping and population increase will be able to accommodate for that. And so the goal of our project was to, first of all, develop a groundwater model for this area where we could simulate groundwater flow and see how it's moving, because we want to be able to see how the nutrients coming out from these cesspools are transported through the groundwater from the high-low divide to the coastline, and then be able to test future scenarios for development needs, manager needs, anything that might come up. So very briefly, this is how the model is developed. You can see the area on the far left panel in the green where my model is located. I focus on the shallow area of the aquifer that's closest to the coastline. And I added all of like the cesspools, and we also have a wastewater treatment plant here, which is highlighted with the red circle. So this is the Kealakehe wastewater treatment plant, and it's located very close to the coastline, as we can see. And there's a lot of nutrients that are added to the groundwater aquifer through this wastewater treatment plant. So we wanted to make sure that we included this in our model. And so in order to make this model, we looked at 4 different parameters that are very important. We looked at the water level, which is the blue panel to the left. We looked at salinity as green, nitrogen concentrations in the red, and phosphorus in orange. And so we decided that we wanted to use 4 different parameters to develop this model, because these are the parameters that we wanted to look at during our future scenario testing. And so it's quite difficult to be able to calibrate the model with so many parameters, because you have to take in so many considerations, and they don't all behave the same way. in the aquifer system. So you do see some scatter in these like plots down here where it's not perfect. We don't have a perfect simulation from our model, but as we know, models aren't perfect. And so we feel that with these 4 different parameters that we use, we got a pretty decent calibration, and we were able to have this robust model that we can look into all of these different things while trying to figure out how the groundwater and nutrients are flowing. And so the salinity, nitrogen, and phosphorus measurements were actually collected by our team members from pumping wells, monitoring wells, and coastal plume samples throughout the basal aquifer. And so these are relatively current measurements that are representing this area. And so once I made that model, I wanted to see where all of these nutrients are coming from, because when we ask what should we do to clean up our aquifers or our groundwater, we always need to know, well, what should we attack first? So I divided up the nutrients into 4 primary sources that I added to my model. So the first is cesspools. As we saw, there are tons of them here. The second is land use, because I added all the different development and agricultural lands to this area, and we add fertilizers. So that's one component. The wastewater treatment plant is a big one. And then the model boundary, because the model doesn't cover the whole island. So we have to assume that there's additional water and nutrients coming in from outside. And so the total nitrogen that the model simulated says that there's about 1,600 kilograms of nitrogen entering the basal aquifer per day, which is shown in this left figure. So you can see the colors show how intense the concentrations are of these nutrients. Of that, the model predicts that about 54% is coming from the cesspools. And so you can see that we have darker concentrations as plumes where the cesspools are located, where all the development is located. So we know that that's a big issue because of how dense this is. Land use doesn't have a huge impact because overall we don't fertilize a lot. Even our agricultural lands, they don't really fertilize. It's all pretty natural. The wastewater treatment plant, relatively, there's only 9% contribution. But if you look really closely at the plume that's coming from the wastewater treatment plant, the concentration is the highest there. So typically everywhere else is like under 2 milligrams per liter of concentration, but then right where the wastewater treatment plant is, we have 25 milligrams per liter, because there's just so much water entering from that one spot. So it's very localized. So it's deceiving to look at just the fraction. You also have to look at the actual concentration that's going in. And then the model boundary, again, it's just a limitation from modeling that you have to assume that they're outside parameters, and approximately 26% would be coming from that. So if we had to discuss like what should we do in the future, we should definitely look at both the cesspools and the wastewater treatment plant. And so if we apply a very basic future scenario to this model, we can assume like development will increase because population's increasing. With higher population, we also need more water. So we need to pump more water. And also, if we have potential climate change impacts, we could expect lower rainfall in this area. So we have less recharge coming into the groundwater aquifer. And so overall, that the model says that it would do what we would expect it to do, is that the water levels would decrease because there's less rainfall, but we're also still pumping just as much, if not more. Because we're pumping so much, we could potentially pump up salt water into our aquifer if we don't have enough recharge to supplement it. And then with more development, we have more wastewater going into the aquifer one way or the other, whether it's cesspool, septic, ATUs. So then that would increase the nutrient concentrations. And so in conclusion, we developed a model for this area using multiple different parameters to calibrate it. We predict that the cesspools do contribute the highest amount of nutrients, but the wastewater treatment plant definitely shouldn't be ignored because it's a very localized spot. And then we could test this to future scenarios. And if you want to read more, you can read this paper. that I had published. And then we also have a follow-up paper that looks at the cost benefits of upgrading cesspools. And that is all. Thank you very much.
Thank you, Brytne, for your talk. Applause from the audience. Are there questions from the audience for Brytne? Please feel free to enter those into the chat. And while we wait for those questions to roll in, I'll get us started here. Brytne, your model is for a specific stretch of coastline on Hawaii Island. Are the results applicable to other places, or would new models need to be created for other locations in order to, you know, come to some sort of conclusion?
I mean, I think overall, it's definitely applicable to other locations, because I think in general, the aquifers at the coastline will behave in similar ways. I would definitely recommend making a new model for a specific, if you have like a very specific location in mind, just because this aquifer is so unique with like that weird jump in head level. Kona also has a very unique climate where we have like a high rainfall belt in one specific area. We have all the volcanoes interacting. So the general ideas I think are applicable, but it's always best to make another model.
Thanks, Brytne. That makes sense that each place has their special things. There are questions coming into the chat. Shuri, would you like to ask your question out loud?
Sure. Great talk, Brytne. Thank you for your talk. I was just wondering, when you talked about phosphorus early on and said it doesn't simulate or model well, why do you think that is?
I think because, well, I think in general, the model might be a bit coarse for the phosphorus measurements. Overall, they're pretty low, but also it's not very stable compared to like nitrogen. It doesn't behave in the same way as nitrogen. And so phosphorus is definitely a huge assumption that's going into model. Some people might even argue that it's not appropriate to model phosphorus the same way you would do it nitrogen. So I think that's really why, it's that it doesn't behave in the same way as like everything else. And so it's not, I would definitely not trust the phosphorus as much as I would trust the nitrogen.
Okay, great. Good to know what your model can and cannot do. Thanks so much, Brytne. There are requests in the chat for links to papers that I'll let you put into the chat as we switch speakers here. Thank you again for your talk. Great job. We're going to bring Devynn to the Zoom stage. And Devynn, we do see your first slide. So whenever you're ready, you can take it away.
Cool. Awesome. I am Devynn Wulstein, as Maya already said. I am a Sea Grant Fellow, and I work with Josh Maiden at the Hawaiʻi Institute of Marine Biology, and also with Thomas Oliver at the Pacific Island Science Center at NOAA. And today, I will be talking about coral reefs, but specifically focusing on translating a theory from the plant world, so taking a leaf out of the plant playbook or the plant book and applying it to coral reefs. So, oh, sorry, gotta click this way. How are coral reefs and forests connected? They're seemingly completely different ecosystems. For instance, one is on land and the other is in the ocean. And so how do we get from a forest to a coral reef? And how do we translate the information that we can learn from forests to that of coral reefs? Well, we can do it through math, and specifically through the theories that govern the ecosystems. And yes, I will be talking a little bit about math, but I promise not too much. Won't be too intense. But these systems are also really heavily affected by intense human impacts, as you just heard. Nutrients and toxins and sediment can come from cesspools and get into coastal ecosystems and can affect coral reefs, causing things like bleaching, And then also we have intense human impacts that create, deforestation and wildfires. And just briefly, kind of for those who aren't super familiar with coral reefs, they're incredibly important for a few reasons. And here's just a few example reasons. They're really useful for fixing carbon. So they take CO2 out of the atmosphere and can convert it into oxygen. And they also allow for habitat and for other organisms to do the same. And they also, as you can kind of see here, it's really colorful. And I tried to add in some fish and other wildlife. They're really heavily diverse in the individuals that they can help maintain in this ecosystem. But for humans, they're really important for dissipating wave energy and providing coastal protection, because waves come through and they break over the coral reefs rather than breaking on land and causing more intense erosion. And specifically in Hawaii and in other island nations and areas, reef fisheries are really, really important for subsistence living, as well as for commercial fisheries or like artisan fisheries. But back to forests. So because forests are a lot easier to study and we don't need oxygen in order to go down and watch what's happening to them and see the devastation that's happened, restoration has been a much larger and much more obvious thing, and something that's been on the forefront of people who work in forefront of the mind of people who work in conservation, simply because we're able to see this, and also because it's much easier to study plants and understand how plants grow and how they die and how they interact with one another, as well as how density of planting these individuals can affect the rates at which they grow and how likely they are to survive. And so ultimately, understanding how, I want to focus specifically on density. So ultimately understanding how the number of individuals or density affects survival is key for restoration and farming and forestry. So kind of this overarching terrestrial idea. And this is a theory that can be applied to coral reefs. And so it's the theory called self-thinning, which describes density-dependent mortality. And so here's a quick diagram of the process. So at an initial time point, these little seedlings have been planted, and they initially start out at a higher density, but over time, they increase in size, but decrease in number of individuals within the same area. And so here's the only math equation. This equation describes this boundary line where N is the number of individuals over a given area. So that's these for trees here. K is a constant value. This is the size with the brackets around it is the average size of individuals in that area. And this alpha term is the power law, or here on this plot, which is a log-log plot and shows a straight line. It's the slope of this boundary line. And so what's most interesting about this is that you can plant trees at a size a little bit less than 5 centimeters around, try to start planting them. And you'll see, and at like 1,000 individuals per hectare. And so as you go over time though, you see that there isn't that much mortality in this population. They're growing in size, but the population density is staying about the same until it hits this boundary line. And so this is a really useful tool for restoration if you want to maintain a certain level of cover or number of individuals. Planting at a certain size can allow, and population density can allow for you to maximize this maintenance of individuals in a population. And so that's where this idea of applying this theory comes for coral reefs, because recently, and I mean, I guess within the past 10 years, about coral restoration has become something of interest, especially in the face of climate change. It's a potential tool for building back populations, as well as trying to maintain the populations. And for the most part, it's done through potentially not super scientifically informed means. And so it would be awesome if we were able to actually apply a theory to build a better restoration plan. And so to do this, we need to see if the self-thinning theory even applies to coral reefs. And to do so, I'm utilizing field data collected through NOAA's National Coral Reef Monitoring Program. And we have approximately 300 to 350 sites throughout the Pacific, where there are multiple time points as well as random surveys. And these surveys are done through structure for motion photogrammetry. So this is an example of what it looks like after the processing has been done, but a swimmer swims over and takes a bunch of pictures continuously, and then they go back to the lab and can build a 3-dimensional model of the coral reef. And then from that, we can extract out number of individuals in a certain area, as well as the size and the morphology and the species, or at least genus level of the individuals. So then we can attempt to assess just this boundary line. Because most of this data doesn't have multiple time points, we're only able to look at this line, not necessarily the trajectory, but we may be able to backtrack out information on those kind of trajectory lines in the future. And so here's an example of all of the data at the transect level, where we calculate the population density of individuals and then calculate the mean radius of those individuals. And so you can kind of see, even though this is just a cartoon line, that there is a boundary that corals seem to come up to with these, the population and size. And so what's also interesting, and particularly interesting for corals, is that there are different morphologies that corals can have and that different species have. And these different morphologies may also describe differences in this boundary line. If we only consider looking at, say, encrusting, which kind of looks like this for some, and you see that it follows a really similar slope to just the everything boundary line, but where the knobby, which you can also see, this is what a knobby may look like, there's a really different slope, and it's much pushed further down. And so these can kind of allow for us to ultimately understand the differences between morphologies and if this self-thinning relationship exists, as well as ultimately attempt to create more plans for maximizing survival and restoration efforts. So, for example, outplanting at the specific density and size in order to maintain coral cover through time. And so by translating from the plant world, which has been able to study their sessile ecosystems for much longer than we have, we can take their hard work and not try to reinvent the wheel, and instead apply those mathematical tools and their empirical theory to coral reefs, as well as both helping restoration, but also for something really interesting, because it's cool to see ecology be translated between terrestrial and the marine environment. And so, yeah, thank you. Any questions? Also, I'm done. Yeah, thanks.
Thank you, Devynn, for your talk. I can hear the applause from the Zoom audience. I know that they're clapping too. If you have questions for Devynn, please put them into the chat box. And while we wait for those questions to come in, I'll ask Devynn, in general, what density and size might be good for future coral restoration, if you were to give us a ballpark?
Sorry, I accidentally muted you there. I meant to unmute myself. Gosh, I think I don't yet have an actual answer for that, but it does seem that, yeah, I can't tell you based on my data, but it does seem like there might be an ideal size, probably around like 3 centimeters in diameter. Maybe that's too big. Diameter. No, I think like an area, a little chunk, not quite microfragmented, but fragmented. But it definitely also depends on the morphology of individuals. And so hopefully that is a question I could more accurately answer or confidently answer in the future. But yeah,
We'll
thanks.
look forward to those results. I can imagine there's so many groups who would be excited to apply these research results in a real way for various coral restoration efforts happening here in Hawaii. There is a question in the chat from Jason. Jason, would you like to ask your question out loud?
Yeah, sure. I was just curious what factors determined, it seemed like that power law changed on the log graph depending on what the value of alpha was. So I was curious what kinds of things, maybe like environmental or other factors that changed that value.
That's a great question, JT. And hello. So actually, it's really interesting that that power law changes, that the alpha changes, because in the terrestrial world, at least for the most part, there is a pretty consistent value of -1/2 for that alpha value. And the intercept seems to be the thing that changes. But that being said, there are the potential for morphology to change that alpha value because sort of the reason why it's occurring is we're assuming a volumetric growth, so that all sizes are the same, so that height is the same as width pretty much, and that's how it's growing. And so if that assumption is invalid, then the alpha might be different. But it's also really interesting for the alpha to be different in this situation. So yeah, thank you. That's a great question.
Okay, we do have time for one more question, and I do see Lillian's hand raised. Lillian, do you want to ask your question?
Yeah. Can you hear me?
We can.
Okay. Great job, Devin. I'm very interested in this. My question relates kind of to the data set that you're using. So it seems like it's from all over the Pacific, and it's a very large data set with these photogrammetry images. And so I didn't know like how more or less those kinds of reefs and contexts, how you think those apply, or if they apply well to coral restoration contexts, which are usually operating in reefs that have much less mature coral growth and maybe are less resilient because of like a lack of, coral herbivores or something along those lines. So I didn't know if you had any intention or the ability to try and ground truth some of these relationships that you're finding experimentally or by working with ongoing restoration efforts, so that you could see how well your results apply to those kinds of settings.
Yeah, thank you. I am planning on, I have completed a pilot study with Montipora capitata, a pretty common.
Donnell.
Oh, whoops, I think her audio went through. A pretty common
Join the meeting.
Hawaiʻi species. And it would be really interesting though, to actually look at more reefs and try to do a little bit more ground truthing. I do have an undergrad who her thesis for graduating is focused a bit on studying how density affects different vital rates through a meta-analysis and systematic review. And so she's trying to address some of those questions with the data that's been published previously. But then, yeah, I'm not exactly sure if it's totally possible within the time frame. It would be cool to do a bit more with it. But on your point of a lot of restoration is focused a bit on the maybe not as many herbivores or kind of a more or a less mature reef. We do actually have, because there are so many data points, there are definitely reefs that have been measured that do show a not mature reef, or one that hasn't reached kind of the closure point or like a climax population. And so we might be able to backtrack out some of that. And this data set also has a lot of environmental information. So yes, and maybe, and hopefully, I think are my summarizing answers. So thanks, Lillian.
I like that. Thank you, Devin. Thanks again for your talk. Great job. We're going to bring Casey to the Zoom stage now. And Casey, when you're ready, you can start to share your screen.
Is it sharing?
We can see.
I tried.
There we go. We see the
There we
first
go.
slide. That looks really good.
Okay, great. Awesome. All right. Hi, everyone. My name is Casey McKenzie. I am a second-year master's student at UH Manoa in the Earth and Planetary Sciences Department. I'm studying under the advisement of Dr. Henrietta Dulai and Dr. Kim Polinsky. And the focus on my focus is on using water I sample from coastal springs to study contaminant source tracing within groundwater. So starting with where the groundwater discharges into our coasts or the coastal springs, and then backtracking from there to hone in on which sources contribute man-made waste to the groundwater. Where do I go next? There we go. To get us started, I'm going to review a bit about what might first come to our minds when we think of the term wastewater, and one way it impairs the integrity of our delicate coastal coral reef ecosystems. And then I'm going to jump into what coastal springs are and a term we use in science to describe them, which is SGD. We'll get more into that in a moment. This will include a little bit on the water cycle and how groundwater flow paths carry contaminants to our coastal springs. I'll conclude with how all this leads into building the goals for my research project, how I hunt for these springs and sample from them, a tidbit on some preliminary results, and then what my expected outcomes mean for our future. So when we hear the term wastewater, one picture that might first pop up in our minds is that of brown water flooding the beach after a big storm. And we know brown water contamination is often an environmental as well as human health concern, especially following major rain events when brown water discharge is amplified. But wastewater actually poses a constant contamination threat to clean water and is just discharged to the sea through a variety of both man-made and natural pathways. Scientists have identified multiple types of contaminants in wastewater, including pharmaceuticals, pesticides and herbicides, detergents, other chemical cleaners, and sewage. These contaminants pose risks to human health, both for recreational users and our coastal zones, and to those that consume other marine life as well. Contaminated water also poses detrimental risks to our reefs. Wastewater carrying excess nutrients from heavy loads of nitrogen-based fertilizers and organic waste from upstream make their way to coastal zones, promoting excess algal growth in reef ecosystems that end up smothering and slowing the growth of corals. And here we see just that. Here's some coral colonies heavily covered by overgrown or overfed algae. This specific image was taken in Kaneohe Bay, and it really does illustrate a common issue in our reefs statewide. But surface water carried in streams and other runoff is not the only source of unfiltered wastewater that makes its way to our coastal zones. Submarine groundwater discharge, or SGD, accounts for and serves as a constant source of nutrient-loaded fresh water straight from our aquifers that emerges at our coasts as brackish coastal springs. So how exactly does submarine groundwater discharge come to be? Here's a short overview of the water cycle and how we understand the development of SGD. We know that our surface environments are the first to experience the interaction from rainfall. Mountains and lakes, streams, soil, vegetation, it all absorbs a portion of the rainfall. But the excess rainfall that discharges to the ocean through the streams and runoff, it either discharges from the surface or it saturates the earth and recharges our groundwater. Because the recharged groundwater first had to have passed through the terrestrial environment before it absorbed into the ground, this water directly interacts with potential contaminants in urbanized and natural landscapes. So now this contaminated fresh groundwater eventually mixes with saline groundwater near coastal zones and exits through the seafloor in the form of SGD, or what most of us would likely recognize as coastal springs. So now if we reimagine that flow path I just described and reimagine it as a delivery system that carries water from higher elevations to lower elevations, we remember that rainfall penetrates the earth until the time it discharges as groundwater to our coasts through springs. We get something like this. And we're going to see a flow path that lies under soils that deliver nutrients from natural soils and other livestock waste. We'll see fertilizers and herbicides from landscaping practices, detergents and commercial industry, caffeine and pharmaceuticals in our waste, and our sewage effluent all along this flow line. And it all infiltrates coastal waters via SGD. This is the flow path I'm aiming to identify or pick apart by examining groundwater contaminants in the SGD. I'll do this by sampling directly at the coastline that lies directly downstream of multiple land use categories. So a little bit more on those land use categories. This illustration depicts the presence of multiple land uses within a single watershed, like golf courses, resorts, and other known areas with a high volume of cesspools. These types of land uses are all known contributors of nutrient waste that infiltrate groundwater, as well as other sources of contaminant classes like herbicides, pharmaceuticals, and those commercial and industrial cleaning products. Hence, one of my project goals is to determine groundwater flow paths that lie beneath this scheme of multiple land use categories. Since there are so many different sources that produce nutrient contamination, like the excess nitrogen from landscaping fertilizers, the waste and sewage, or just natural waste, I'm aiming to pick apart which sources could most likely be responsible for the excess nutrients by identifying other contaminants that also ride along in the groundwater from those same sources. So I'm specifically testing for 11 different chemicals in my samples, and they include BPA and alkylphenol. These are chemical agents that are common in industrial cleaners and detergents and could identify leaky sewer lines. Pharmaceuticals, including ibuprofen, acetaminophen, caffeine, carbamazepine, and sulfamethoxazole, are ingredients that are present in household sewage effluent and could hence identify the presence of cesspool leakage to groundwater. And then lastly, I'll be examining ingredients from pesticides and herbicides like pyrethroids, glyphosate, atrazine, and 2,4-D, which could serve as tracers for landscape practices that are a source of fertilizer infiltration to groundwater. So how do I hunt for coastal springs or SGD? Fortunately, these springs leave a thermal signature when they emerge into our coastal zones. So like you can see in the thermal imagery map in the image on the right, here we see SGD in the blue and in the green shades, and it's seeping into the coastline with a slightly cooler signature as opposed to the warmer seawater we see in the red and orange hues. Typically, the springs are at best visible at low tide, as it trickles protruding out of the sand, but in some cases they can be rather obvious, like in this video that I'll play for just a couple seconds. Here we can see the SGD bubbling up right from underneath the sand at the edge of the beach. It's playing again, sorry. So since sampling SGD can be kind of tricky, there are a few pieces of equipment I use to collect water from the springs. This includes a battery pack that helps me run a pump that's connected to a hollow push point rod with some Tygon tubing. And you can see that in the left image. You can kind of see the push point sampler. Here's the pump and then the tubing pulling the water out from beneath the sediments. And before I decide on where to sample, I have to survey beaches visually while also cross-referencing my location to the thermal imagery maps like the one I showed in the previous slide. I then test the water I find seeping from the sediments with a YSI device. This device helps to check the salinity. And if the salinity reading is low, much lower than seawater, then I push the sampling rod into the sand and set up the pump. This helps collect the SGD before it is diluted by seawater from any incoming waves. Hence, if I sample at a low tide, the springs are easier to find and I run less risk of encountering waves that dilute my samples. This is important because I want to analyze my SGD samples for contamination that occurs before it mixes with the seawater. So the less salty the spring, the better contaminant signature I can get. And lastly, I'm going to just touch on some preliminary results. I know it's a pretty busy screen, but for each figure, I have the bottom axis is just measuring a value for my chosen contaminants or nutrients that I'm looking at. And then the left axis correlates with the latitude of where I sampled that spring. So if I were to show these figures next to a map, you might be able to see better along a north and south running coastline where the samples line up along that coastline and other land use sources, but essentially running north and south. And the values you see in the left are measurements of apparent nitrate and phosphate. I'm really trying to key in on the nitrate. So although the levels are significantly elevated in coastal springs due to high nutrient loads to the coastal zone, there are only a few obvious locations of elevated patterns. So as such, nutrient concentrations alone are not enough to determine exact sources, which is why I didn't touch on this really at all, but I will also be looking at stable isotope values of the nutrients within the groundwater as well, which is our middle figure. And that's, I'm hoping to use these values to try to pick apart more so of the types of wastewater sources versus just the presence. And then lastly, in the right two slides, here's some preliminary data from some pharmaceuticals I've tested in the water, ibuprofen and sulfamethoxazole. And you can see ibuprofen is showing some really strong signatures in a couple areas where I've sampled, and this high persistency, I'm hoping that maybe this might be a really good signal or a tracer for identifying those precise contaminant sources. So overall, the big picture, my overall research goal is to use those chemical signatures that I am able to identify in the SGD to trace the major nutrient contamination sources. And with this information, I hope to develop eventually a pollution scoring index that could help others in the future determine contaminant sources and overall help land managers narrow down which practices might need some fine tuning. These efforts can not only help us improve our groundwater quality, but it could also reduce contamination risks to our valuable yet delicate coral reef ecosystems. And I'd really like to thank Sea Grant for your support in pursuing this educational research opportunity, and especially my advisors for their infinite guidance and expertise, as well as our friends at the Nature Conservancy. We have coordinated with them extensively in our study sites to help identify springs and communicate with local land managers and just really get a know-how and an understanding of the area, and slowly learning a lot about the study region for my project. And that wraps it up.
Thank you, Casey, for your
Thank you.
talk. Thank you so much. There are already questions in the chat, which is wonderful. We'll have Yoko ask his question first, and then we will go to Saren. Yoko, do you want to ask your question out loud?
Yeah, gladly. Awesome presentation, Casey. Great job.
Thank you.
My question is, does atmospheric deposition of nitrogen play a significant role in Hawaii?
That is a really good question. And I haven't spent a lot of time reading into atmospheric deposition. I want to say no. I feel like the deposition is really so much more sourced by land and natural events like on the surface and within biological roles in the geology. But that is something definitely that I want to read more into.
Thank you.
Thank you.
Okay, let's go to Saren's question. Saren, would you like to ask your question out loud?
Sure. Yeah, I was just wondering if you had any idea about how many, like approximately, or any clue how many SGDs do you think there are along coastlines of like Oahu or any of Hawaii?
Oh, that's a really good question. I mean, when I walk across beaches, I've seen it on North Shore, almost most places you go, but it's not always obvious. My study site is off of the South Kahala coast of the Big Island, so mostly near Waikoloa and Puoko. I sampled roughly 50 sites within an 8-mile stretch of coastline, and I know that I was definitely not getting all of them. It's really hard to pick them apart, but using those thermal infrared imagery maps really helps hone in on where they're at. It's not necessarily that there's just like one spring that's obvious. It could be that there's a portion of coastline that's showing a digital signature or a thermal signature. And those springs are just little trickles out of the sand in tiny little spots along the coastline. So identifying just one spring versus a coastline of lots of seepage, it's hard to quantify. That's definitely another goal of my project, is hopefully maybe if I sampled several different spots on the same beach, I might see different isotopic signatures in the water. And that could help delineate if it's kind of one major flow path versus several smaller ones, but I don't have a direct answer for exactly how many.
Okay. Yeah, and then on your 8-mile stretch, were some of them like much more prolific than others? Like some are really small and some are really big, or?
Most of them are really small. The one that I showed bubbling out of the surface, that I only saw, I think, 2 out of the whole coastline, 2 or 3 locations that actually it was bubbling up. Most other locations, it's literally just like a little constant trickle, and you have to search or even dig a little bit to get a YSI in the water enough to even see if it's got freshwater content.
That's pretty cool.
Yeah.
Thank you.
Yeah.
Thanks, audience, for those questions. What I'd like to do now is I'm going to bring both Brytne and Devynn back to the Zoom stage. And the way I do that is I add the spotlights. And now what I'd like to do is we can open up the Q&A for any of our speakers. So if you have questions for any of the graduate researchers that gave talks today, please enter them into the chat, or you can raise your hand in your Zoom box and we can make sure to get your question answered. So while we wait for those questions to come in, I will ask our graduate researchers, maybe we can go round robin. What is your experience been like conducting this place-based research? Tell us something that you've learned along the way as you conducted research in a community setting. We can start with Brytne and we'll make our way around here.
I think the biggest learning experience is how doing place-based research is not on the same time scale as like a grant. Because you have to make so many contacts with people like landowners, there's to contact well owners, and you have to apply for different permits all the time, and that can't always be done within a 2-year time frame. So it's definitely hard in that sense, and it's something that should be considered a little bit more by the grant agencies.
That's a great point, Brytne, that building relationships takes time. That's a really important point. Devynn, you're dealing with 300 to 350 different sites in your work. How do you stay grounded in place and community as you conduct this research?
Luckily, I've had a decent amount of opportunities. It has been a bit tricky with COVID to really stay grounded and not just over Zoom. But I am part of MOLSA, which is the Moku'alo'e student and graduate student organization. And we have just done and restarted one of our Malama Aina days. And we're going to start doing them every semester. And it was a really awesome experience to actually get dirty and really put in some sweat and effort and take out mangroves and plant native plants around the Hawaii Institute of Marine Biology. And so I guess for me, it has been hard because my data is everywhere to try to be focused in place. But thank you, Sea Grant, for providing the opportunities. And I think, yeah, if you do go find opportunities, they're out there. You just have to be willing to put in the work and get a little bit sweaty sometimes, I think, too. So, yeah.
I like that, Devynn. Call to action to get a little sweaty sometimes. I like that a lot. Casey, do you want to close us out here? Anything you've learned from conducting research in a community setting, conducting research in places?
I feel like it's kind of a combination of Brytne and Devynn, especially with my experience having to work with folks on another island amidst a pandemic. So it's challenging both coordinating when we can meet, especially, you know, for me, it was, I had to do it at low tide. So I had a tiny window every month and it didn't always work. And then also bridging those gaps, you know, talking to landowners and people, you know, they don't know you and you don't know them. And you want to make sure you communicate that you're just here to learn and try to help everyone. So it definitely, it's a time investment, reaching out to people way ahead of time and being clear with availability and goals and making sure that everyone's on the same page with similar goals. That way everyone can work together on it.
Thank you for that. Yeah, sounds like investing time into communicating and building relationships has been really important. Thank you again to all of our speakers. Great talks today. I can feel the applause from the rest of the Zoom audience. Thank you so much. This is our last Science by the Sea seminar for 2021. We'll keep you posted on our plans for 2022. We're super appreciative that you came to watch today, and we'll see you sometime. Happy New Year. Thanks, everyone.
Thank you.